Sathyabama Institute of Science and Technology.
Good morning, everybody. This is Jesia Persis Preethi from the Department of Biotechnology, Sathyabama Institute of Science and Technology, deemed to be university. So, in the previous class, we were discussing about DNA replication. In the current one, we will see the differences of replication that takes place in prokaryotic and eukaryotic cells. So, first and foremost, just having a quick recap of what is DNA replication. Now, there are a lot of enzymes which come into action. First and foremost, if you see the DNA polymerases, they help in the process of replication, and the direction of replication is from 3' to 5' end, meaning which the daughter strand which is formed is from the 5' to 3' end. So, since it is semi-conservative process, there are two copies of replication which is formed, two copies of DNA. One is leading and the other one is lagging strand. And we also discussed about Okazaki fragments, which are discontinuous fragments which are formed on the lagging strand, which are being sealed by the DNA ligases. The DNA helicase is the one which opens the DNA primer. DNA helicase is the one which opens the replication fork, and here the DNA polymerase 3 comes into action where it splits apart and where it holds the strands apart from one another and starts replicating it. And prokaryotic replication takes place in the cytoplasm of the cell. The reason is they do not have a very well formed nucleus. Therefore, since it doesn't have a nuclear membrane, the genome is directly present in the cytoplasm itself. So, therefore, the replication happens inside the cytoplasm itself, and each DNA molecule has only one single place of origin, and the replication process happens in bidirectional. So, two replication forks are created. So, therefore, the replication copy forms, and there is one replicon in bacterial chromosome. Polymerase 1 and 3 plays a predominant role in this procedure, and the pieces of Okazaki are generally very huge pieces. So, they range somewhere between 1000 to 2000 nucleotides, and it is a very fast process, meaning which out 2000 nucleotides are formed for a single second, and DNA is double stranded and circular DNA. Therefore, this is how the replication takes place in prokaryotes. Whereas eukaryotes has a well-defined nucleus with a nuclear membrane. Therefore, the replication takes place in the nucleus of the cell. And single DNA molecule has several sites of origin which employs the proteins for the single-stranded binding proteins and other enzymes for replication to take place simultaneously. So, at one point of time, there could be many replications taking place simultaneously, and therefore, we can see multiple replication bubbles in the process, and there are approximately around 50,000 replicons on the eukaryotic chromosome, and DNA polymerase is responsible for the process, and we have DNA gyrase also is necessary for the process. The Okazaki fragments are tiny, unlike your prokaryotic where there are large Okazaki fragments. The Okazaki fragments are tiny, somewhere measuring between 100 to 200 nucleotides in length, and it is very time consuming process, and hundreds of nucleotides are inserted every single second. The DNA structure, if you see, it is double stranded and linear DNA that is present in the eukaryotic replication. So, here is a pictorial representation for better understanding. So, the DNA replication in prokaryotes, you can see circular DNA, therefore a single replication origin with the replication fork, where the daughter strands are formed in the center on the either pair of parent strands, and the repliosome assembles a replication fork and slides along and synthesizes the DNA. And we also said the Okazaki fragments are very huge. The reason is this. And if you see the eukaryotic DNA, it is double stranded and linear. Therefore, at single point of time there could be multiple origins of replications, and we also said that it has shorter Okazaki fragments. The reason is as the replication forks open alone, the sliding of the DNA polymerases and synthesis of a lagging strand takes place. Therefore, they have shorter Okazaki fragments. Thank you.